acting on effector proteins, right? So what would be those 3 types of effector proteins? Metabolic enzymes is one. So we've got one.
Transcription, regulatory proteins.
Transcription.
Yeah,
cytoskeleton. Yeah. So those are 3 proteins that can be acted on by signaling proteins, right? So thank you guys for filling that in. So these, once they're acted upon, they can work long or short distances in the body, right? So what is that called if I want to say in a more professional way, long or short distance?
Contact dependent or paracrine signaling.
Okay, so paracrine signaling is what I'm looking for. So paracrine would be what?
Short and fast.
Short?
Fast over short distances.
Fast, right? And then endocrine would be?
Slow.
Cool. So who can give me an example for paracrine? There's so many right answers for this one.
There's one where it's the local mediator
Okay.
that gets dispersed out, and then it's within a short vicinity that all the other cells receive it.
So that's a great example. Local mediator is like a cell adjacent to a cell. So this is like in the epithelial lining of the gut or of the brain barrier, and the cells are just releasing talking cues next to each other, right? Okay. What about another paracrine signaling example?
Neurons.
Neurons, right? So that's a really good example because neurons can be really long. They're one of the longest things in our body. They go from your cranial nerve through your spine all the way down your arm to your leg, right? So that seems long-ish, right? But for the most part, paracrine signaling with neurons, synaptic-wise, is really short. It's almost cell to cell. So I know she skipped out a lot of neuron material for you guys, but let's say here is our cell body, axon, and then our dendrite ends. Remember action potentials? So let's say an action potential starts, flows down, and then we get the release of acetylcholine, right? That gets released from this neurotransmitter. What could this be released onto? Acetylcholine. We talked about it, right? In lectures, a lot of examples for acetylcholine.
Muscle cell?
Muscle. So muscle is a great example of why this is short and fast, right? So let's say here's a small muscle in your finger. So this happened pretty close to the speed of light, right? So like take your finger, move it, that's acetylcholine in this exact paracrine signaling pathway, right? So that's the type of example I want you guys to be thinking about when we're talking about these things, because if you can visualize it and see it like that, then you know it has value, right? This isn't just because the textbook and Dr. Banner said so. So now let's talk about long distance. So we used neurons as paracrine, but neurons, like I said, run through the entire body, could also be long distance. But besides neurons, what's another example of long distance endocrine signaling besides neurons?
Hormones?
Hormones, great one. Anything else? So let's go off hormones, right? So how would a hormone be long distance, right? Where's it secreted from? Let's say the adrenal glands. You're running from a lion 10,000 years ago, right? Your tribe. Lion's chasing you guys. Adrenaline is in your blood, so that's, I like to think about these things in a realistic way. So that would be endocrine, slower. And the reason it's slower is because this is not being secreted cell to cell. So it has to build up in the blood to have a certain concentration to start binding to all those receptors. But the problem is with things like hormones, once they bind, they stay bound for a long time. So that fight or flight feeling that we experience today, you can experience when you sit down to take this test that we're going to have coming up, right? Those stay in you for a long time and have more of a consequence long term for health effects. Just know that that's what they're associated with, and that's all you need to know for the exam there. So they are inactive When? When would these be inactive? When GTP?
GDP.
GTP?
D.
I'm just kidding. Okay, you guys got it. It could be a test question. She might try to throw you off of them. I don't know what you thought about the last test, if there was any trick questions. She swears she doesn't do trick questions, but... GPCRs. The first model organism, you guys don't need to know this, that was discovered, you know, how we found out about GPCRs, desensitization, was the sea slug, not a common model organism. So they would electrocute the tail end of the sea slug and it would have a response, you know, acetylcholine, boom. Eventually, after enough of that low stimulation, it didn't have a response. And they discovered they could desensitize the stimulus, and that was through a GPCR pathway. And this opened up so many windows for neuroscientists and molecular biologists, but that was kind of the end of the fame as far as the sea slug goes. He doesn't get pushed into many studies anymore. Pretty cool though. So as far as desensitization, you can't always have these receptors on and ready. That transmembrane protein, then you've got that unit part of it, right? So you've got your little receptor, ligand, they bind, and then your alpha does what?
It makes a conformational change.
Conformational changes occurs, yeah. But then what leads the GPCR to go signal further downstream inside the cell?
Beta and gamma.
Which one? So yeah, your theta, gamma. So this is a huge part of drug targeting. That's why I asked you if you talked about drugs. Maybe she'll get into it, maybe she just cut it out. But for pharmacology, this has got to be 80% of drug targets we're looking at is what GPCR can we bind to. Problem with this though is there's a lot of side effects.